map of regulatory elements in regulatory interaction in human genome. As all you know, the networks of gene regulation is very complex. To understand this complexity, it's really important to understand the regulation of transcription factor and binding of transcription factor. Besides all of the local binding of transcription factor, three-dimensional structure and three-dimensional interaction is very important to understand the complexity. Numerous genomics technology have been evolved to understand and study three-dimensional structure of chromatin and chromatin conformation. Among those, 3C, 4C, 5C, and high C are the ones that widely used in recent studies. Virtually all of the experimental methods are based on cross-linking the chromatin, shearing the chromatin, either with sonication or using restriction enzyme, and cleavage of the DNA, as I mentioned, and proximity ligation to make a chimeric fragment of DNA, and then align it back to the genome to find the interaction between two parts of the DNA. Among those technology, high C allows us to detect genome-wide map of the interactions. But basically, the limitation with these techniques is the low resolution of the technique. So these techniques allows us to detect the interaction in the resolution of the 20 to 50 kb. And for achieving a very high resolution map, we need to have a very, very deep sequencing. So it requires very deep sequencing to get a resolution less than 10 kb. We chose to use CHIAPET chromatin interaction analysis by pair-end sequencing. This technique reduces the sequencing depth that requires to map the interaction, and actually reduces the depth of the sequencing and brings up the resolution of the techniques by targeting the factor of interest. So that's why we chose this technique, and what we got in our data, it was the resolution around 3 kb. I brought a simple flowchart of CHIAPET workflow, just in case you're not familiar with the method. It's based on cross-linking the chromatin, shearing the chromatin, and then pull down with factor of interest. And then we ligated linkers in two end of the fragments. These linkers had a recognition site for MME1 enzyme and also is biotinated. Then in a very diluted reaction, we do proximity ligation and then digest with MME enzyme, and then we have a fragments around 80 kb. Then we do pull down with streptoavidin beads, and then parent, basically library prep and parent sequencing. Okay, so what can be addressed with CHIAPET? What we were looking for? What factors and combination of factors are involved in establishing and maintaining the chromatin structure? That was the main question. Also, how do the chromatin states of distal interacting site affect gene expression? And also, how do these interactions regulate tissue-specific gene regulation and function? The other question was that if this interaction kind of used to regulate a different biological process or not. Here is the data. We started to compare our data with the DNA's hypersensitive side and see how much coverage we could get with our experimental method. We used 6 broadly distributed factors for histone marks and PAL2 and RAD21. And by that, the binding peaks from these 6 factors, we could cover almost 80% of DNA's hypersensitive site, which was actually very high coverage. And also, we covered 82% of our annotated regulatory element defined by ENCODE, and that was including 99% of TSAs and 98% of enhancers. So then we look at a TAD domain, which was defined earlier by high-C technology, and see if the CHIAPET interactions falls within a same TAD domain or across the TAD domain. Most of the CHIAPET interactions, 97% of CHIAPET interactions connect two regions within the same TAD domains. So it was a kind of confirmation of the proposal by other investigators about some subcompartment of the chromatin, which they call a TAD as a active and kind of basically transcription factory of the chromatin. So then we compare our data and interaction data with 5C. 5C is very high resolution techniques to detect chromatin interaction, but the limit of these techniques is basically low coverage of the genome. It's almost 1% or more. But it's very high resolution, so we wanted to confirm our interaction and compare it with high C. We got almost above 60% of CHIAPET overlap between CHIAPET interaction with high C interactions in all of our data set. Here I brought a globine leucos as an example. This is the leucos that is known for non-interaction, and you could see a good basically overlap and coverage of CHIAPET interaction with 5C interactions. And when we compared basically our data set with a earlier basically produced data set with the same factor, we actually got around 40% higher coverage and overlap with 5C. So the quality of data was a little bit improved in our hand. So then we look at the profile of interaction. What we have in the interacting loci, we use ChIP-seq data from 102 transcription factors and intersected with CHIAPET data. And we saw that we have enrichment of 4 factors, CTCF, RAD21, and SMC3, and ZNF143. So we believe that these are 4 factors that are very important to maintaining the long distance interaction in genome. And then we look at the strengths of interaction with calculated Z-score, and we found out that when we have RAD21 in both sides of interaction, we have a stronger long distance interactions. So RAD21 basically plays an important role in those kind of interactions. Then, again, we use ChIP-seq data and CHIAPET data and look at proximal binding and distal binding. And again, these 4 factors pop up, and SMC3 and RAD21, basically component of cohesin complex, CTCF and ZNF143, which is a transcription activator, basically are enriched in distal targets. Means that where we have the basically transcription factor in a distal site that connecting to a parameter in our interacting site. So again, kind of confirmation on the role of these 4 factors in maintaining long distance interactions. We look at a combination of the factors in the interacting loci, and also basically specifically looking at the hot region, basically high occupancy transcription factor binding regions. We found out that we have enrichment for the hot regions, and those hot regions tend to interact with each other, and that's kind of support again the transcription factory model that was earlier proposed by other investigator. Okay, here actually I want to show you that different biological process regulated with different network of proximal or distal transcription binding. In the basically the column, there are the list of transcription factors, and in the row are biological process. And basically here we have the ratio of p-value proximal versus on distal binding. The red ones are the spots that are greater, we have greater enrichment in distal target. And the blue is where we have enrichment in proximal binding, means we have the binding of transcription factor in a promoter. So what it was interesting to see was that a lot of biological processes that maintain the housekeeping function of the cells are regulated through proximal binding and proximal network, and a lot of basically dynamic process of the cell are regulated through distal binding, like cellular response to a stimulus, or basically cellular response to stimulus, any stimulus like drug or any immune response. So they are regulated through distal binding. Then we look at functional impact of interaction and between promoters and distal regulatory elements. Using RNA-seq data, we actually find that wherever we have a promoter connecting to a repressor or any weak enhancer, we have less gene expression, and wherever we have a disconnection between any enhancer or transcribe region, these are the basically annotation based on the encode again. Between the enhancer and transcribe region, we have more gene expression. So that was actually kind of expected, but that was again a kind of the first global report on functional impact of interaction on gene expression. On the other side of the slide, I want to show you that different these interactions are kind of cell-specific. Here, wherever we have a promoter connecting to different types of the chromatin, and look at a K562 specific gene and broadly expressed gene, when we compare those, we see that the connection between enhancer and promoter are the most cell-specific interaction that we could see. So we have more gene cell-specific gene expression when we have a connection between promoter and enhancer rather than other type of the chromatin states. Then we want to see that those interactions are kind of cell-specific or not. We compared two data sets from GM K562 and also GM78 cell line, which is LCL lymphoblastoid cell line. And then using a method called DBS scan, we found that around 1,000 specific interactions for these two cell lines, And then we look at the specificity of, Then we look at the specificity of an interaction and the effect on specificity of basically cell-specific gene regulation. So wherever we have the genes that whose promoter overlap with the end of the cell line specific interaction, we have more significantly higher level of expression in that cell line. You could see in the basically here in the middle graph in K562, basically we have more gene expression when we look at basically cell-specific interaction of K562. So they are functional. Basically, these cell-specific interaction are functional. Other thing that we look at, we basically experiment, it was the type of the interaction. We wanted to see that which type of interaction are more specific. Again, we look at promoters that connect are connected to different states of the chromatin, and we saw that connection between promoter and enhancer are basically cell-specific interaction, long-distance interactions. Okay, summary of my work. Actually, we used 6 broadly distributed factors for histone marks, for active histone marks, PAL2 and also RAD21, and we kind of cover 80% of DNA's hypersensitive site, and we cover almost 99% of TSS and 98% of enhancers. The medium size of interacting loci was around 3 kb. That's the resolution of the basically method, 3 kb, and the distance between 2 interacting sites was around 120K. Majority of ChIA-PET interactions falls in the same topological domain, and strength of the interaction was correlated with presence of RAD21, in both sides of the interacting loci. RAD21, SMC3, ZNF143, and CTCF are enriched interacting loci. Hot region was enriched, and they tend to interact with each other. And also, TF regulate different class of biological process with basically proximal or distal target, as I mentioned earlier. Many GO terms were mediated by proximal binding, basically was involved in general housekeeping function of the genes, and many GO terms that was regulated with distal network. TF network was more involved in a dynamic process like signal transduction, immune system process, or response to any drug or stimulus. Chromatin states of distal interacting region have important impact on gene regulation. As I show you, wherever we had a promoter connecting to enhancer or transcribe region, we got more transcription, more gene expression, and wherever we had a connection between a promoter with repressor, we got a suppression in gene expression. Enhancers are highly cell-specific compared to other type of regulatory elements, and the interaction between promoter and enhancer where the most specific interaction happened in the cell. So basically cell-specific interactions. Okay, sorry. The acknowledgement doesn't look like acknowledgement here, sorry about that, but I would like to thank my PI, Mike Schneider, and also my colleague, Doug Ponchel, that he analyzed most of the data, and the rest of the lab and co-workers helped me to do this project and also finding. Thank you so much.
Again, I encourage students and postdocs and trainees to come and ask questions.
Yeah, I would like to ask a question. Thank you, I find this very interesting.
Sure.
You're surely aware of one of the first ChIA-PET papers, this Lee et al. In Cell, which also published such a study on a genome-wide map of ChIA-PET regulatory interactions. I'm just wondering what is different in your map to this map published 2 years ago?
Okay, you're talking about the GIS the work from GIS group in the cell publication. So
exactly.
they actually used just PAL2, so they just map promoters and basically connecting regions, which was enhancer, and also it was just one factor, and basically it was around, I guess, 30% or something coverage of DNA's hypersensitivity side. They didn't measure, we measured, we want to compare it with our result, and our basically coverage, it was more or less than what we got in this study.
And how do both studies overlap?
Actually,
I mean, do you find these interactions again that they find?
No, the type of data analysis was very significantly different. What we used for our basically finding the interaction, it was distance corrected method. So what we have found from 6 factors is what around 30,000 interactions. But for them, for one factor, actually, they found a way more like 60,000 or so, but because they didn't consider any basically distance correction in their method. So yeah, we have some basically overlap, of course, but we believe that our analysis method was more accurate, basically considering the distance correction, and that's why when we basically compare it with 5C data, we do see higher coverage with 5C data rather than their method. So yeah, there is some overlap, but we got some improvement in the analysis and also experimental part.
Thank you.
Sure.
You've shown that housekeeping genes are regulated by a different set of transcription factors than genes which are developmentally regulated. But I wonder, is it possible to analyze your data to look for the differences between transcription factor binding enrichment in the proximal and distal region? Are they enriched in the same transcription factor binding or different, possibly? When you look at the promoter, the proximal, and the distal enhancer region, are both ends enriched in the same transcription factor binding or different? Is it possible to look at?
Yeah, actually, let me back, if I understand your question correctly. Here we tried actually to see if those transcription factors are in a proximal binding, direct target means the proximal binding, and indirect target means the basically distal binding. And we saw these 4 factors was enriched in basically all of the data set. But if you want to look at a specific transcription factor, I think it's just doable. And we can just, looking for a transcription factor. Sure.
Okay, thank you. We're going to move on.